Click the bell icon and subscribe to our channel so that you will not miss any classes. Hello, welcome to class. In the previous class, we have discussed in detail about the vectors and the criteria for the selection of vectors. Now, we shall understand what are the types of vectors available in biotechnological works. Let us take up them one by one. The first type of vectors available are plasmids. Plasmids are, as we know, they are the circular double-stranded DNA capable of undergoing self-replication in bacteria. So naturally they are found in bacteria, but these plasmids have been modified and they are altered in order to use in biotechnological works. And we shall take up the two examples of plasmid vectors, which are popularly used in biotechnology. One is Plasmid Ppr 322, and this is the first vector which is constructed, and it is full form as plasmid. P here refers to plasmid and Br is the scientist name. The two scientists are involved in constructing this vector. They are Bolivar and Rodriguez. And this PBR 322 structure will be like, as we know, plasmids all of them are circular double-stranded DNA molecules, and for diagrammatic representation we will write like this, but naturally when they are found in bacterial cell they will be like this. So just have an imagination. Okay, so they will not be in this form. It is just to write in a diagrammatic representation. And here, as we studied from the previous, any vector for using in biotechnology, they should have importantly three things. One is selectable markers, origin of replication, and the multiple cloning sites. So this PBR 322 also have all these structures. It has selectable marker genes as the antibiotic resistant genes. One is ampicillin resistant gene and tetracycline resistant gene. That is, two selectable marker genes are present. These two are the selectable marker genes. And of course, multiple cloning sites are present. There are two cloning sites present on tetracycline resistant gene. One is BAMH1 and SAL1. And likewise, on ampicillin resistant genes, two selectable marker genes are present. One is PVU1 and PST1. And apart from this, there are other cloning sites present like EcoR1, Clavin, and Hint3. So these are the available restriction sites. And remember, on PBR 322, it has the cloning sites or the restriction sites for 20 different restriction enzymes, and I have written here whichever are very important. And apart from this, it has a special sequence called the rope sequence, and this rope sequence will code for proteins which are necessary for replication. So here the rope sequence is very important for controlling the copy number. The rope will produce the proteins which can bind to origin of replication and bring about the successful replication, which will result in a high copy number. And also, as we we discussed, it should have the origin of replication, and it has here the ORI site, origin of replication. So these are the important aspects which are present in PBR 322, which is the extensively used plasmid in biotechnological works. And apart from this, there is other plasmid vector used, that is called PUC18 vector, and here P refers to plasmid, and here it is developed in University of California. It is developed in University of California, and 18 is the plasmid number it has just given. And here the difference between these two are, it has the selectable marker genes, selectable markers as LAC-Z. We have discussed the alternative selectable markers, right? So it has LAC-Z, which is responsible for producing beta-galactosidase enzyme, and all we have studied. And here it has another antibiotic resistant gene, that is ampicillin resistant gene, as a selectable marker. Both are present. Antibiotic resistant gene is also present, and alternative selectable marker LAC-Z is also present. And here these two will have a different size, and the size of PBR 322 is of about 4363 base pairs, and of PUC18 is, it is quite little, 2688 base pairs. And remember, the size of a vector should be less so that it can propagate easily in the host cell. If the size of vector is too large, then it is difficult to maintain in the host. And one more thing is, if the size of vector is too large, then how can we insert the gene of our interest? For this, we have to attach the gene of our interest. So for that, always the vectors should have as much as less size. Okay, so this is the first type of vectors available, that is the plasmids. And apart from this, the second type of vectors used are bacteriophages. Bacteriophages are the virus which can infect bacteria. They can infect the bacteria, just they send their genetic material into the bacteria, and that genetic material will get integrated into the genome of the bacteria and make the bacteria to produce the proteins and the other things which are responsible for the replication of virus. That is the basic thing. By exploiting this fact, scientists have thought we can use the bacteriophages to introduce the gene of our interest into the bacterial cell. On that course, there are two important bacteriophages which are commonly used as vectors. One is lambda-phage. Lambda-phage is a bacteriophage having 48.5 kilobase pair long double-stranded DNA as genetic material. And you should know about the diversity in virus. There are different sorts of virus which have single-stranded DNA as genetic material, double-stranded DNA as genetic material, single-stranded RNA as genetic material, as well double-stranded RNA as their genetic material. So here there are different kinds of virus, but here the lambda-phage is one which is having double-stranded DNA as its genetic material, and a size of about 48.5 kilobase pairs. So just have imagination, this is the length of the bacteriophage DNA, and here is the double strand. If you look here, I am writing a single strand on both the sides because here the small length of DNA is single strand, that is of about 12 base pairs. 12 base pairs on each end. 12 base pairs on each end of lambda phage DNA are being single strand. Single strand. And very importantly, they are complementary. They have complementary base pairs, and you know that what is complementary. Suppose here it is having G, P, C, A sequence like this, and here it will have a sequence like C, G, A, C. The sequence will be like this. If it is complementary, they can base pairs easily. And this site, it will form a site, right? So this site, which is complementary and capable of undergoing complementary base pairing, the site of bacteriophage is called cos site. So this cos site is very important in packing the viral DNA into the phage head. So the bacteriophages, they have the head and collar and the tail, and genetic material will be packed here. If it is a long length, then that long length cannot be accommodated easily in the head. If it is have a cos site, they can undergo complementary base pairing, so it will become shorter, so that it can pack up in the bacteriophage head easily, and this will be necessary for easy introduction of the genome into the bacteria. So here, so this can be used in biotechnological work for transferring the gene. So here the cos site is remained as such, then the DNA will be treated with any restriction enzyme of our interest, and then the gene of interest will be introduced here, and because of the cos site, the DNA will get packed into the phage head easily. Then the phage head is made, or the bacteriophage is made to enter or made to infect into the bacterial cell, so that the further things will be as we know. And the second type of bacteriophage which is used as a vector is B, I will write it as M18. And this M18 is popularly known as Plasmid. So these things will be important for competitive exams, so you should remember them. M18 bacteriophage is used as a vector, and this is commonly known as Plasmid. And this have the genetic material of length 6407 base pairs, which is having much smaller size than the lambda phage, and it is single stranded DNA. It has double stranded DNA as genetic material, it has single stranded DNA as its genetic material, and it can infect E. coli cells. Can infect easily the E. coli cells, and when it gets infected into the E. coli cells, then it will be converted into double strand. Suppose it is E. coli cells, here it will converted into double strand, and this double strand is capable of undergoing replication in Escherichia coli cells. Hence, the double strand which is formed in the Escherichia coli cells is called the replicative form. Replicative form, or we can refer as RF, refers to replicative form. And this replicative form can be isolated and manipulated. Can be isolated and manipulated for use in biotechnological works. And it is possible to isolate the single strand from here also, and it is popularly used in gene mapping or knowing the sequence of any genetic material. And also it is possible to clone the DNA of our interest into the bacterial cells. So this is the second type of vector, that is bacteriophage as a vector. And now the third type, and one more thing here you need to remember. These, that is the plasmids and bacteriophages, can transfer the DNA of our interest, the length less than 10 kilobase pairs. That means they can transfer the DNA but of not too much length. The length is restricted here. Any gene which is having the size less than 10 kilobase pairs, they can transfer effectively. But there are genes which are having the size more than this. What about them? For that, they have developed the alternative vectors also. Let us discuss them in detail now. The third type of vector available is Plasmids. Plasmids are the hybrid vectors. Hybrid means we refer to the term which is having the combination of two. So it is a hybrid vector which is constructed by taking the characteristics of plasmid and lambda phage. So this is artificially constructed vector, so which can transfer the long length of DNA. So here they have took the characteristics like ori site, selectable markers, and the restriction site from plasmid, and they have took the cause site. We have discussed just now, the cause site is important for packing the genetic material into the phage head, and that site is complementary, so that site is taken, and the combination of these two will give rise to the plasmid. It is just like the plasmid will be taken and a cut made here, and then through that cut they will clone the cause site. So then it will become a plasmid. So wherever the selectable markers and the restriction sites are present, there we can transfer the gene of our interest, and it can be transferred into the bacterial cells. And this vector, that is plasmid, can transfer or can clone the DNA of about length 45 kilobase pairs. We have discussed previously, plasmids and bacteriophages can clone the DNA of length less than 10 kilobase pairs. So just look at the efficiency of this. So it can clone about the length of 45 kilobase pairs. And the fourth type of vector are the shuttle vectors. So here the shuttle vectors are constructed. They are also a type of plasmids, we can say. So here the shuttle vectors are created or they are made in such a way that they can clone the DNA to bacteria as well as yeast. They can clone the DNA into bacterial cells as well as yeast cells. Bacteria are prokaryotes, yeast are eukaryotes. So they are made in such a way that they can successfully transfer the genetic material into bacterial cell as well as to the yeast cell. For bacteria, the basic necessities we know, that is selectable markers are present, Ori site is present, restriction sites are present. But for the replication in the yeast or in eukaryotic cell, there are some additionals required. Telomeric sequence is required, telomeric sequence, and the centromere. So these two are additionally added to the plasmids. So then it will become a shuttle vector. Let us discuss the one mostly studied shuttle vector, type of shuttle vector later. But for now, yeast episomal plasmids. Episomal plasmids are the type of or the examples of shuttle vector. Just remember, the shuttle vectors are the vectors which are made to replicate or to clone the DNA into both prokaryotes as well as eukaryotes. That is the basic thing you should know. Then the fifth type of vector available are the bacterial artificial vectors. So these vectors are commonly popularly known as BAC. The bacterial artificial chromosomes are constructed or they are made on the basis of F plasmid of bacteria. In bacteria, there are F plasmids present, and these F plasmids can transfer from one bacterial cell to another bacterial cell during the process of conjugation. During the process of conjugation, they can transfer. This is F plasmid, and they can transfer into the bacterial cell, and they can transfer the genetic material from here to here. This is the natural thing. And here the scientists have made a bacterial artificial chromosome in such a way that the plasmid will have the gene coding for F factor. The gene coding for F factor is Ppr. The Ppr genes are responsible for coding the F factor, and plasmids are inserted with, this is a plasmid, and they will be inserted with the, as we know, Ori site, selectable marker which will have, and multiple cloning sites for various restriction enzymes, and along with this it also have Ppr genes. So if the Ppr gene is present, this plasmid can enter from one bacteria into another bacteria very easily. Okay, so this is the basis of constructing the bacterial artificial chromosomes. So wherever the multiple cloning sites are present, there we can clone the gene of our interest, and this is made to transfer into the bacterial cells. So they can transfer, that means this bacterial artificial chromosomes can transfer the DNA of about length more than the 45, that is up to 100 kilobase pairs it can transfer, and these are used in genomic projects. In genomic projects like Human Genome Project, HGP. So Human Genome Project, they make use of bacterial artificial chromosomes that can clone the DNA of long length. If the long length is cloned, so that it will be easy for the mapping of the genes. Okay. Then the sixth type of vector used are yeast artificial chromosomes. Yeast artificial chromosomes, popularly known as YAC. So here it is a kind of shuttle vector. Yeast artificial chromosomes are kind of shuttle vector. We have studied here, right? So which means here it is a plasmid which is made or which is constructed in such a way that it can replicate both in bacteria as well as yeast. So for that it is having the requirements like it has the oocytes for both bacteria as well as yeast. So that means it has two oocytes. One oocyte is for yeast and one oocyte is for bacteria. So when it is entered into bacterial cell, this oocyte will work. When it enters into yeast cell, this oocyte will work. Okay, and also the selectable markers for two types of selectable markers, one for bacteria and one for yeast. I write it as selectable markers. Here refers to selectable markers, and selectable marker for yeast and selectable marker for bacteria. And of course, the multiple cloning sites are present, and this is common for both bacteria as well as yeast. For any restriction enzyme, they act here, and the gene of your interest will be cloned here, and it will be transformed. Okay, so this is the shuttle vector. So which is can clone the DNA to either to the bacterial cell or to the yeast cell. And don't think one can clone at a time to both. At a time it is not possible. One can clone to bacterial cell or to the yeast cell. So that is the condition. So this type, that is the yeast artificial chromosomes, can clone the DNA of a maximum length. So that is up to 100 to 1000 kilobase pairs. This much long DNA can be cloned. And remember, so yeast artificial chromosomes are the one which are capable of cloning the DNA of maximum length. So remaining all will be having less capacity, but the maximum capacity is for yeast artificial chromosome, which is one of the type of shuttle vectors. So these are the different types of vectors which are available for cloning the DNA to bacterial cells or to the yeast cells. And in the next class, we shall discuss the vectors available for cloning the genes into plant cells and the animal cells. Here we have not discussed about the plant cells and animal cells. It is possible to clone the DNA to plant cells and animal cells also. Okay, so that we shall discuss in the next class.